Um, Hello, everyone. Thank you all for joining in for Dr. Tapinos' seminar today, hosted by the Center for Computational Molecular Biology. My name is Heon Huang, and I'm a second-year PhD student in the Computational Biology program. Dr. Tapinos is my primary co-advisor, and we're all very excited to have him with us today. Dr. Tapinos received his MD and PhD in molecular biology from the National University of Athens, Greece, and completed his postdoctoral training at the Rockefeller University, where he was introduced to the field of molecular neuroscience. And there he studied how pathogenic leprosy bacteria regulate glial cells of the peripheral nervous system. And following his postdoctoral training, he joined the faculty at Geisinger Clinic, and an opportunity to join the Brown faculty prompted his departure from Geisinger Clinic in 2016. And he is currently the director of the Cancer Epigenetics and Plasticity Lab and Molecular Neuroscience and Neuro-Oncology Research at Rhode Island Hospital, and is also an associate professor in the departments of neurosurgery and neuroscience here at Brown. So without any further delay, please join me in welcoming Dr. Tapinos. And I guess we're all over soon, so you may start whenever you're ready.
Yeah, thank you very much. Thank you, Sharon, for the introduction. And I'm very glad to be here and giving this talk to CCMB. And let me share my screen. Okay. So let's start with the science. So the title of my talk today is Cancer Stem Cell Plasticity, Integrating Computational Biology with the Bedside. And the reason I put together this kind of title is because this is exactly what we do. I'm not a computational biology, but I admire and I computational biology all my life, and I understand some parts of it. Let's put it this way. And I think it's very important to actually use computational biology to understand very important cellular processes. And what I think I can play a role is to bring the knowledge that we create with computational biology and computational science to the bedside and to make better treatments for patients. So in this respect, I will give you an introduction for the disease that we're dealing with, right? So the disease is brain tumors. And what I'm showing here, I'm showing you, maybe let me get the pointer, okay? So I'm showing you like an MRI from a patient, and what you see here is a glioblastoma, which is the grade 4 very aggressive brain tumor. What happens in a patient, and this is actually a very typical history of a patient. So it comes to the clinic, the tumor is diagnosed, they operate post-surgery. You see this white area where the tumor is, this is edema. But a month later, there's nothing there. It's just basically the cavity where the tumor was. So everything is removed. So everything looks good 1 month after the surgery. And the patient with radiation and chemotherapy, 7 months later, has a tremendous, huge tumor which recurs in the brain. And in some instances, it's a different area of the brain or even the different hemisphere of the brain. And that is actually deadly. So after millions and billions of dollars spent the last 50 years for research on brain tumors. And basically, we have tried everything: immunotherapy, radiation therapy, obviously surgery, different surgical techniques. 20 years ago, when I was studying medicine, you know, I was reading cases where people did total hemispherectomy. So they were removing half of the brain to deal with this disease. And the disease was coming back to the other hemisphere. So nobody really can treat these tumors. And to give you some data from the National Cancer Institute and National Brain Tumor Society, so the mean survival rate for glioblastoma is 12 to 18 months right now. Still, the 5-year survival rate is less than 6%. And what this means, that means that children with leukemia, 9 of 10 children now survive for 5 years, which is actually a time frame that we think that, okay, that we are actually cured the disease, most probably. For adults with breast cancer, 9 of 10 patients with breast cancer, if it's diagnosed in an early stage, they can survive 5 years and above. With brain tumors, adults, less than 2 people every 10 people can survive 5 years. For children with brain tumors, is less than 1. It basically never happens. Why this is happening? And from a lot of research that has been done during the years from different labs and from my lab, so we know that the cells that actually provide this aggressiveness to this disease, these are cancer stem cells. So years ago, the Parada lab at Sloan Kettering actually proved that the neural stem cells is the center of origin for these tumors, where you have some oncogenic driver mutations happening at some point in the life of these neural stem cells. And with these mutations, these become cancer stem cells, obviously. From there, and from the first oncogenic mutations, everything else depends on epigenetics. So basically, after that, you have asymmetric divisions, further epigenetic changes, epithelial mesenchymal kind of transition, although these cells are never epithelial, let's call it a mesenchymal transition, transdifferentiation to stem-like cells, which they form these tumors, which, as I explained before, it has all kinds of cells within the environment of the tumor. What I mean all kinds of cells is that cells that carry the founder cancer stem cells that carry the parental mutations. There are cells that have epigenetics, which are called epigenetic subclones, because they have a genetic mutation, which is actually induced by epigenetic factors. There's genetic subclones, which have different mutations that can happen as well. And then you have cells with different chromatin changes and different methylation changes. And what is not included in this is that we know that there are cells with different RNA changes, like in the methylation of RNA, non-coding RNA. And this bigger circle here actually tells you, and believe it or not, that this is a patient of this is a tumor of one patient. All the things can happen at once in one tumor. So the heterogeneity is incredible. So recently in 2019, in this Cell paper, because we thought that by patient, every patient has different, characteristics. So that is the heterogeneity that we're dealing with, which is already incredible. But now we know that within the tumor, there is intratumoral heterogeneity. And if you do a single cell RNA sequencing of this, in these tumors, you see that the cells, based on this analysis, this is Mario Suva's paper, that they cluster the cells in 4 different clusters. And they call them the mesenchymal cluster, mesenchymal like 1 or 2, but this is one big cluster, the astrocytic-like cells, the oligodendrocyte progenitor cells, and the neural progenitor cell-like cluster. So there are 4 different clusters. And what happens is that the cells within this cluster, based on epigenetic factors and microenvironment factors, they actually cross clusters. So these clusters are not stable. They are actually, cells are always in motion. And what makes these cells differentiate or de-differentiate in different different phenotypes? Now we know that it's reprogramming transcription factors, epigenetic histomodifications, expression of non-coding RNA, other microenvironment cues like hypoxia, inflammation. It's also what we do as treatment-induced stress if we do radiation or chemotherapy, and also the rearrangement of chromatin structure, genome architecture changes on cells, push them into different phenotypes. So this, all these concepts and all these properties of these very plastic cells brought this idea to my mind some years ago, that maybe some of you or all of you have seen this. This is the epigenetic landscape of Waddington's epigenetic landscape that was published in 1957, where he actually described fate decisions of cells, which he made this landscape. They say when they're in the top and they want to go to different valleys, there are some equations that actually can tell you where the cell will go and what is where they're going to end up. And this is the epigenetic landscape that was, as I said, published in 1957. Now we can say that epigenetic factors actually change this, the trajectories of the cells and the fate decisions. And we believe that states that we believe that before that are terminal states, now they are not, and cells can go back and acquire different phenotypes. And that is a classic epigenetic landscape in cancer. So what I thought that this exactly what we should be studying in this disease. We should understand what drives the decisions of cells to change phenotypes in a very complex microenvironment as the brain. And not only what drives them, but in every state of the cell and in every stage of the cell, can we identify the key factors, the drivers that actually make the cell to make a decision and become an intermediate cell and progenitor cell and different kind of cellular phenotypes. And then if we manage to do that, basically we can actually use this, this model here and model the states of the cells in, and this is quite simplistic, but just so to understand is we can actually think that a cell in a network state between A and B, and it expresses genes that actually make them most probably be in A, because these genes are highly expressed versus the B state, or both A and B states are equally expressed, or the B state is more highly expressed, or both states are very highly expressed. So if you put these probabilities in an epigenetic landscape, then the cell can be, down here where you have a stable position where one network is highly expressed, or it can be up there where both networks are very highly expressed, so that you have a lot of changes. And you could imagine that this cell has, if you want to call it in biophysics terms, and I will show you what I mean, it has maybe higher free energy, maybe higher entropy. So the question becomes, how do we calculate, how do we understand what each cell does in cancer, in brain cancer? So we decided to do exactly that. So we started a project, and Heion is here. This is part of Heion's graduate program, David's graduate program, thesis program as well. And Ritambra is helping us, obviously. And we also have a collaborator, Natalie, in Israel. She's a Caltech graduate biophysicist that helps us on that. And the idea is to build the epigenetic landscape of brain tumors where we have cells and we monitor in a time experiment from day 0 to let's say day 10. In different days, we take the cells in a single cell state, and initial state, we call the initial state is the initial culturing of the cells. By the way, these are all cells coming from a human patient, from the brain of the tumor, from the brain of a patient with a tumor. We have states where cells can do whatever they want, basically, based on the environment, which are called high entropy states. And we may have states, and I say maybe, because we don't know if the cells will ever enter a terminal state. But in order to include them, we have cells, let's say, that they acquire a terminal state and they cannot do anything else. So then we expose these cells into differentiation-inducing media, which is the easiest way to induce differentiation. And in the future, we will also put them under hypoxic conditions. We now have a chamber to do that, or with chemotherapeutics like temozolomide, which is a gold standard therapy right now, or under radiation, and actually take single cells, do single cell multi-omic sequencing at different time points. And multi-omic, we're doing RNA sequencing and ATAC sequencing right now. And then with the help of Natalie, we're doing what is called free energy barcoding. Basically, she can actually assign free energy by doing free energy calculations, every single cell, we can actually calculate the free energy of every single cell and then relay the free energy to the transcriptome and the the epigenetics of the cell, and also calculate single cell entropy. Using machine learning and cell state prediction, we can actually predict what the cell will do, which state will acquire after the continuous treatment. And then we will do something like what I call perturbation network analysis, but there are also way more sophisticated ideas developed by Heion, Ritambra, and David right now. So, but to give you an example, what I mean by perturbation analysis, this is an experiment that we have done actually in not in single cell, but in bulk ATAC-seq. And I'm putting this schematic as well as the epigenetic landscape because networks are important. So what we did here, we took glioma stem cells and we did an ATAC-seq. Again, this is like not single cell, this is bulk. And then we identified all the transcription factors, which are we call the founder transcription factors. What are these transcription factors? These are transcription factors that regulate genes, so they form a gene network, but also the gene network regulates the promoter of these transcription factors, so there is a balance between expression of the genes and the regulator. So these transcription factors, genome-wide in glioma stem cells, were 100 transcription factors that have this ability. So we call them these stable founder transcription factors. And then using a machine learning approach, we start perturbing the network, basically taking away each individual transcription factor and seeing which, if we take 1, 2, 3, 30, 40, 70, whatever transcription factors, which one induces the highest perturbation on the network and makes the network unstable. So, and we got some very interesting data. The interesting data were that even if, and we call them offsprings are the networks that are produced after you removing each individual transcription factor. So in most cases, the transcription factors, if you remove them, the network doesn't change. So basically, and you see it here with the green bars. So basically, these are transcription factors that actually don't do really anything. They're just expressed, right? And these the master regulators, these are called bystander transcription factors. And actually, I have to make a comment here that we usually see these transcription factors in a lot of our experiments, RNA-seq data or publications. And in reality, these transcription factors are just there because they're always there. It's not the ones that are driving your phenotype. What drives your phenotype is these little guys here with the red, which if you take them away, it completely destabilizes the whole gene expression network. So something similar, I'm not saying we're going to use the same approach, but something similar we want to do for every gene and expression and identify master regulators that actually dictate phenotypic states in this cell. So why we want to do that? Our goal is basically in a therapeutic kind of thing to develop cell state restriction as an epigenetic therapy for glioblastoma. So what is cell state restriction? If we find the major critical players that actually dictate how a cell transitions from states, and we take them away with therapies, with new drugs, right? Then the cell cannot do exactly that, cannot transition in states, so it remains in one phenotypic state. This cell is very easy to target. This cell is very easy to take away. So that's in my opinion, that's the way to deal with the cancer that actually adapts constantly. So far, I called it a pipeline. So my lab has developed these very exciting options to do exactly that. One is an antibody against a very highly expressed protein. This was done with collaboration with Jack Elias's lab. The second is an enhancer RNA, antisense oligonucleotides targeting an enhancer RNA. The third one is novel small molecules, which were producing de novo inhibitors targeting histone deacetylase 7, which we have done all the work to prove that the role that it plays. And the last one is inhibitors, which are also de novo new compounds that we designed in the lab. And these are completely new drugs that are targeting the M6A RNA epitranscriptome, which is actually post-transcriptional modification mechanism of the RNA that regulates major genes that define cancer stem cells. So I want to show you some data now and share with you some data which are for the first option, the chitinase 3-like-1 antibody, because this is we have data to actually prove how we are blocking, exactly restricting phase transitions with this antibody. So chitinase 3-like-1 is one of the most highly expressed proteins in glioblastoma. This also, it's a secreted protein, it circulates in the serum of glioblastoma patients. And it correlates with decreased overall survival in patients with glioblastoma. And we know that in glioma cells from publication, it promotes invasion, survival, malignant transformation, but we don't really know what it does in glioblastoma. So when, when we saw this kind of expression, you see like 6,000 reads in glioma stem cells, almost nothing in the normal brain. And these are different glioma stem cells for different patients. In TCGA, very highly expressed and very, very significant correlation with low survival. So we decided to study what is the function of chitinase 3 like 1 in glioblastoma. So if you do a clustering of high expressing glioblastoma, chitinase 3 like 1 in glioma stem cells, and you cluster them with a group of signature genes, which are called the mesenchymal signature genes in glioblastoma. These were published like almost 10 years ago by Verhaak in a Nature paper. You see that the high expression of chitinase 3 like 1 correlates with the mesenchymal signature, which is the very aggressive and actually stemness factors that give aggressiveness to the cells. And this is actually, if you take now chitinase 3 like 1 expression and put it into the Mario Suva single cell RNA sequencing data, you see that with red, the high chitinase 3 like 1 expressing cells, this is single cell RNA-seq data, are actually here, down here, which are the mesenchymal kind of cells. But because the cells always transition, you see them also in the astrocytic and even in between. But it's not really expressed in the neuron progenitor cells or the oligodendrocytic. In our hands, we took like isolated glioma stem cells from 9 patients that came here to Rhode Island Hospital. And we see that chitinase 3 like 1 is expressed highly in all of them. And then what we did, we took recombinant chitinase 3 like 1 and we said, okay, let's put it in the media of these cancer stem cells. And 48 hours later or 72 hours later, we actually went and did a FACS analysis of the cells. And we saw that just by adding recombinant chitinase 3 like 1 in the media as a secreted factor in the environment of the cells start changing phenotype. They start losing expression of markers like SOX2 and CD133, which are actually proneural and markers that are not expressed in this mesenchymal glioblastoma, and starting activating expression of mesenchymal markers. This is a remarkable switch. These are proteins. And this is to happen within 3 days, just by adding a recombinant protein in the media. This is really something important. So we said, okay, we have to understand how this protein works. So we did a single cell RNA sequencing after adding chitinase 3 like 1 in the media of these cells. And I'm showing you the pipeline of how we deal with this data. So this is a glioma stem cell from one patient where we formed first the Jacquard resampling to identify stable clusters for these cells. And when you see the clusters superimpose the control and the chitinase 3 like 1 treated cells. And a second patient looks like this. Just, I don't even need to explain to tell you how different 2 patients with glioblastoma, that's the same disease, but the cells are completely different and they are clustering completely different. So, but no matter what, you can do a functional classification of these clusters and you see that there are clusters with, for example, cells which are actually very highly expressing mesenchymal markers or cycling, like these clusters here, or you have the hedgehog signaling with green here and here, which are all, these are aggressive pathways for these cells, right? So then in order to understand the contribution of chitinase 3 like 1 or to see if the chitinase 3 like 1 contributes in phase transitions of the cells, we actually employed RNA velocity. RNA velocity is a method where it calculates the ratio between spliced RNA to unspliced RNA and infers the movement of cells because transcripts are moving from a pre-mRNA to mRNA. And you can vectorize this movement within the clusters of these cells. And as you see in these 2 patients, if you do RNA velocity, you see that there are certain clusters of cells, and I will give you an example for cluster 13. In the control, you see the vectors go one direction. When you add chitinase 3 like 1, the cells actually completely change direction. And the same happens with cluster 5. In the second patient, we have cells moving from cluster 1 and 4, not actually moving in control, but it actually start moving very rapidly after treatment with chitinase 3 like 1. So there is definitely phase transitions of the cells. So the question is, what drives these transitions? In order to understand this, because every cluster and every cell, that's the beauty of the single cell RNA-seq, it has a transcriptome associated with it. So you can go back and say, okay, what is cluster 5 and cluster 13? Let's get the genes that are expressed there. And we rank these genes based on percentile change. And here you see for example, to identify the top driver genes, we impose a cutoff that we say, okay, genes that are changing from almost 0 to more than 75, these are the top drivers. And this is percentile change. And then we actually have the names of these genes. And if you see, these genes are actually the most important mesenchymal drivers for the cells. So we are proving here that a recombinant protein in the environment of the cell, this is not a transfection, we're not overexpress the cell, we just add it to the environment of the cell, make the cell completely change its transcriptome and move, at least some of the cell, move to a more mesenchymal, more aggressive phenotype. And then we wanted to see if there's movement between clusters. And with the help of Ritambra, we actually developed this method where we calculate transition probabilities, cluster to cluster transition probabilities. And with this method, we calculate what is the probability of cells to move from one cluster to the other by comparing the movement of each cluster to all the other clusters. Let's put it this way, so we understand, or at least I understand. So then, in order to when we did that, so in one patient we saw that there is significant movement between clusters 1, 7, 3, for example, which are the mesenchymal again clusters and the cycling clusters. And the second patient, there was movement towards cluster 4, but mostly towards cluster 3, as you see here, which again, these are the cycling and MYC targeting clusters. By the way, MYC, the transcription factor, is one of the most important drivers of glioblastoma. So we are showing again here that a recombinant extracellular protein, which is very abundant in the environment of this tumor, pushes the cells to acquire more aggressive phenotype and more invasive even phenotype. And we wanted to calculate if this is a transient phenotypic change or these cells acquire stable characteristics, which actually will go against what we thought initially, that these cells are completely plastic and can keep changing. So in order to research that, and you see here, this is cell-cell transition probabilities in all the clusters. These lines are they're showing the path of each cell, where the probability of each cell starting from one state where it will end up. And you can actually calculate this by running a Markov chain modeling to actually tell you where the cell is going to go. And if it goes somewhere, is this a terminal state, or the cell keeps the ability to move from this state? And then you can calculate this from every single cell. And by doing the Markov chain modeling, you can calculate significance between control and chitinase 3 like 1 treatment. And what we see in both cases, and you see with very high significance, 10 to the minus 16, is that when you add chitinase 3 like 1, actually cells will not acquire a terminal state, which means that the cells become very capable of changing and transitioning to different phenotypic states. This is exactly proves the hypothesis that we put forward, that there are microenvironmental factors, and in this case, chitinase 3 like 1, I'm sure there are others, that actually pushing the cells to keep constantly changing phenotypes. And what drives this in terms of transcription, right? Because this is the genes. So in order to understand this, we did this ATAC sequencing of these cells. And in areas of high accessibility, by running 3 different programs, they all programs came down to one transcription factor being the very highly active and regulated. And this is MAZ transcription factor. I'm showing you here the footprint, the actual footprint of the transcription factor binding in the genome in the chitinase 3-like-1 treated cells, more significant than in control. So in order to see, is it really the genes that actually, and the processes that actually regulate this transition depend on MAZ transcription factor, we inhibited transcription, this MAZ, and you see this inhibition is very, very significant by siRNA. And what we see here, that the genes that are up and down, and the upregulated and downregulated genes, this is RNA sequencing, which is here, we did the footprint on these genes to show which one of these genes have actual MAZ binding on promoters of these genes. By doing this analysis, we found that MAZ regulates the genes that are NOTCH1 dependent and hedgehog dependent after chitinase treatment. And that is exactly what I show. If you remember one of the first slides, when you add chitinase 3-like-1 to the cells, you are activating pathways of hedgehog signaling, NOTCH1 signaling, and mesenchymal signaling. So we are finding that MAZ is a transcription factor intermediate that actually mediates these phenotypic transitions of these cells. So to go to the clinic, because my title says going to the bedside, so we collaborated with Jack's lab, and they developed this mouse and humanized monoclonal antibody against chitinase 3-like-1. So we took this antibody and we did this experiment. We isolated the patient-derived glioma stem cells. We implanted these glioma stem cells into the brain of immunocompromised mice in day 0. 1 month later, we added a small little pump in the mice, which was filled with the inhibitory antibody, and a catheter in the brain giving this antibody to the tumor cells. And then another month after the treatment, we did an MRI on these animals to see what we're doing. Are we doing something by inhibiting chitinase 3-like-1 specifically? Does it have an effect in the disease? And not only it has an effect, here is the segmented MRIs of the animals. We are actually inhibiting glioblastoma growth by 65%. And that is incredible. And, you know, in order to actually link this data with what I'm showing you all this time about RNA sequencing, we repeated this whole experiment, and we actually surgically removed the tumors, the treated tumors versus the non-treated tumors. And we did a transcriptomic analysis to identify which genes are inhibited if we use an antibody to block chitinase 3-like-1. And I'm showing you here the genes. These genes are the same genes that chitinase 3-like-1 activates in single-cell RNA-seq data. So this is a really, really important and specific therapy. And that is actually if you remember the mesenchymal signature that I showed you from Verhaak, we're inhibiting the mesenchymal signature with the antibody in vivo. So starting from a very, very basic biology study, if chitinase 3-like-1 has an effect in the phenotypic transition of these cells, we actually have now a potential therapy against glioblastoma. And I know that Jack's lab and we're working towards this to develop this for human clinical trials eventually. And this is very exciting. And this is one of the projects. The same work is happening with all the other pipeline of therapies that we're developing, where we are studying this in a very basic level, and we're trying to understand mechanisms that push these cells in different phenotypes with the hope that we will have a continuous pipeline of new drugs and new treatments for this disease that can be used in combination with current treatments that are trying to kill the cells, or these new therapies together to help these patients actually deal with this disease in a better way. And I will stop here. I'm happy to answer questions. And I just want to acknowledge all the people in my lab with incredible work and collaborators, the Department of Neurosurgery here, collaborators from helping us with different projects. and funding from Warren Alpert Foundation, the National Cancer Institute, Brown University, and the Department of Neurosurgery. And thank you very much.
Thank you, Nikos, for the wonderful talk.
Stop sharing. Oh, I don't know. Oh, it's paused. Okay.
Do we have any questions from the audience?
I have a question, Nikos. This
Yes,
is Anita Norman.
Yeah, hi, Anita.
This is really great stuff. It's amazing. So how much have you done or have you done any comparisons of the probabilities, or maybe other people have done it, the probabilities of these different phenotypes in adults versus children? Has that been studied very much?
Yes, we are not comparing directly adult versus children, but we know that for children, for pediatric glioblastoma, right? So the pediatric glioblastoma has some different features, and the different features are they are not so much heterogeneity, because the pediatric glioblastoma develops mostly because there is a mutation in histone 3.3 variant. These cells, which kind of locks these cells into a certain epigenetic state. So what we are seeing is that I just glanced through it. I roughly mentioned this treatment that we're developing for an epitranscriptomic based therapy, where we're targeting an enzyme against RNA methylation. This treatment is very effective on pediatric glioblastoma as well.
I see.
Chitinase 3 like one, for example, what I spent this talk to show, it doesn't really work this way in pediatric glioblastoma. So there are differences, and also the other big difference for DIPG is that's why it's called the diffuse intrinsic pontine glioma. It grows in the pons, which is very difficult to treat because of the area.
I see. Interesting. Thank you.
And then Pinar, your question.
Thank you for the great talk. I am more of a computational methods type of person, so I was curious about the Markov chain modeling.
Yeah.
From my understanding, the transition probabilities are already inferred before that step. Yes. And then there you're inferring whether a state is terminal or not, right?
Yes, yes.
How do you, I guess I have difficulty imagining how the Markov chain modeling helps with that if you already know which states are transitioning into which states.
But you don't. What you don't know, I understand your question. What you don't, you know what exactly you said. Yes, you do know this. But what you don't know, let's say state 1 and state 2, right? And you know that we have shown that there is transition probabilities from state 1 to state 2. The question is, okay, a cell goes to state 2. Is it going to stay there? Is it a terminal state for this cell or not?
But if you are already confident in your transition probabilities, you would also have numbers on probabilities from transitioning, like from state 2 to another state, right? So do you update your transition probabilities during the?
They have to, because if you start, let's say you start to define states, right? So, or not states, actually that's not true. You start to define clusters of cells. So there are a certain number of clusters in each, a certain number of cells in each cluster. So it's not that the cells that go from 1 to 2, now they make 2. There are already cells in 2. So there are cells that will leave 2 and go to another state, for sure. But you don't know if it's the cell that came from 1 to 2 will stay into 2 or will go to another state, right?
So does that mean some states will be terminal for certain types of cells, but
yes,
for other
yes,
types of cells?
yes,
Okay,
yes.
I see. Okay, that's very interesting. Okay, thank you.
Do we have any other questions from the audience? Okay, I guess I'll just ask one. We know that there is a lot of heterogeneity in patients with glioblastoma, and with the heterogeneity, I imagine it's really difficult to try to get like a general solution, a general like approach, therapeutic approach for this. How do you think we should go about dealing with the heterogeneity in our data? Do you think like we should go towards a more personalized medicine approach or something along those lines?
I think that for glioblastoma, it's definitely, even though it sounds as a cliche, we're going to do personalized medicine, right? It's at the end of the day, because the heterogeneity is so major in this disease, yes, I think it's a model of a disease that where you must have treatments for different scenarios.
Mhm.
Obviously, I don't believe that every patient will be different and will have a different. But at the end of the day, and this is where again I think the modeling that we're trying to do and the predictions that we're trying to do, if we find, and again, just for the sake of the discussion, if we find like 5 factors that are considered major drivers of these phenotypic transitions, and you have drugs that target these 5 factors, I believe that this kind of scenario will cover 95 or more % of the patients, right? There will be extremes, yes, there will be extremes.
Right.
But at the end of the day, that's what you're doing. Right now, we don't have any of that.
Mhm. Right.
Right now, the mindset of targeting not only brain tumors, any tumor, is kill, 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 right? We need to kill the cells and get rid of this disease.
Mhm.
So, I mean, I was giving a talk one day, and I actually said that, why don't we turn brain tumors into a chronic disease? And people said, "Whoa, you lost your mind? What is going on?" I said, no, what I mean is that we have to give the opportunity to the treatment that will get rid of the tumor to work, because now it doesn't work for sure.
Mhm. Okay, thank you. Pinar, is that a lingering question, or do you just have the hand?
Sorry, I forgot to
Okay.
lower it.
Okay. If we don't have any further questions, I think we can end the session here. Thank you all for joining. Thank you, Nikos, for the wonderful talk.
Thank you.
So thank you.
Yeah. Bye.
Bye.